Welcome to the Northeastern IPM Tick Series webinar. This is the second in the series, and we are delighted to introduce Dr. Stephen Rich, and he's going to be talking about what happens when and if reducing source or preventing tick bites has failed. So Dr. Rich studies vector-borne zoonoses, i.e. those transmissions that are facilitated by blood-feeding arthropods. In particular, he's focused on the study of Lyme disease and human malaria. In the realm of tick-borne diseases, his lab has engaged in first-of-its-kind passive surveillance for established and emerging pathogens in the range of human-biting ticks. And from that work, they're finding the spread of emergent pathogens that in turn drives efforts to learn the causes of that spread. And he's going to be sharing with us about the projects that his lab has been working on today. So I'm delighted to introduce Dr. Rich and grateful for his time in sharing his expertise with us. And so with that, I am going to turn it over to Dr. Rich.
Oh, thank you, Jana. Thank you for the invitation. It's a pleasure to be here. So what I'd like to do today is perhaps a little bit different than other talks, but I think it will tie in with things that you've heard before, for example, from Dr. Kirby Stafford, because what I'd like to do is address the concerns that we have about assessing risk and mitigating the risk of tick-borne diseases. So I have a little video here to play. I'm sorry. I have a little video here to play, which underscores the tick behavior and the thing that we most worry about. So here's a female deer tick. She's doing an activity that she utilizes in order to take her blood meal. She's going to crawl out to the end of this branch and then quest. So we know about this. This is the questing behavior of the adult deer tick. And here's a passerby that's going to fall victim to this questing female deer tick. You see that the tick takes purchase on the individual's skin, finds a suitable place to feed, and then she inserts her mouth parts. And this is the thing that we most are concerned about. This is what we try to avoid ticks getting on us and engaging this kind of feeding behavior, because we know that things are coming out of that tick which are infectious to us. And obviously those are the causes of disease. But what I'd like to do in our time together today is talk about not just preventing that action of the tick getting on you or biting you, but also discuss certain things that we can do even if the efforts that we've done to control the ticks have failed. So we all know that there are things that we can do, personal protection measures that we can do, like proper clothing and certain chemistries that we can use to protect ourselves from tick bites. And we know the importance of checking our bodies for ticks. Some of the personal protections we can use, which Dr. Stafford certainly talked about, are we can treat our clothing, we can also treat our skin, and all of these things can prevent ticks from getting on us in the first place. In addition to personal protections, there are source reduction efforts, and we often think about these as the best approaches, things that can kill questing ticks, kill ticks on hosts, reduce infection, and I'm not going to review those here because they've been addressed very well in other talks that you've heard. But what I'd like to emphasize is that every tick-borne disease is a story, and every story has a beginning, a middle, and an end. And so while we often focus on the pre-tick bite, that aspect of keeping ticks off us, I'd like to remind everyone today that there are things that we can continue to do to mitigate risk of tick-borne diseases, even once the tick bite is occurred, and sensibly even after the tick bite has occurred, because we know that the public health risks associated with tick bites follow this storyline, and there are things that we can do to reduce risk throughout the entirety of that story. And I say this in the context of, depending on your generation, the younger generations, I think we're getting more adept at managing our own public health concerns. So many people spend a lot of time on the web, and we're thinking about the risks of diabetes and heart disease, et cetera. And we have a pretty good idea of how these threats work. We know when we're supposed to change our diet. We know different things that we can do to prevent ourselves from encountering these public health threats. But somehow when it comes to tick-borne diseases, I think we lose some of the image. We think of only those very early stages, that pre-tick bite stage. Like if I just keep ticks away from me, that's all I can concentrate on. What I'd like to encourage us to think about is that there's something that you can continue to do even after those measures have been taken. And so to understand that, let's frame how we go about describing risk of a tick-borne diseases in the context of how a public health person would explain those. So you can think of risk as being, as the slide shows here, the likelihood of a hazard causing harm. And I distinguish that from the hazard itself. So in this case, I'm using sharks just because it's very often sort of the canonical example that's used. But the hazard could be the presence, absence of sharks in the water. And the risk is the likelihood that that hazard actually caused harm to yourself or to some population of people. And, you know, just to take the absurd example, if you're swimming in a freshwater pond, you're probably not encountering the hazards. However, if you're swimming in the ocean, you may be encountering the hazards, but the risk may be relatively low, either because the hazards are infrequent or because you can take measures to prevent that, you know, based on where you swim or those kinds of things. And so what's often forgotten in this process is that it's not just the hazard and the risk that we worry about. What we really are most interested in, more often than not, is the exposure that mitigates that hazard and risk. And so the very simple relationship between them is that the hazard and risk, the overlap of the hazard and risk, is that exposure component. And so taking it from the shark example to a tick example, we can think of the ticks as the hazard and the associated pathogens, and then the risk that we often measure when we look at infectious diseases in people is just the incidence of disease, but it's more often than not the exposure that we're most interested in. And this exposure piece is, interestingly enough, less often measured, and I'd like to tell you how we measure it and why we measure it the way we do. So I'm going to show you the three components of exposure to tick-borne diseases. The first parameter of exposure is the tick species. So it turns out that different tick species pose different hazards. So not all ticks are created equal in this regard. And some of us may already know this. For example, deer ticks are really the only species that transmit Lyme disease. Dog ticks don't transmit it, Lyme disease, and Lone Star ticks don't transmit Lyme disease. However, there are other pathogens that are transmitted by dog ticks and still others that are transmitted by Lone Star ticks. So you see, it's very important to understand what tick species one has encountered or been exposed to understand what risks are presented or what hazards are presented by that tick species. So the first exposure factor is to understand the tick species. And here's just a summary of the different tick species, the three canonical tick species that bite people: deer tick, dog tick, and Lone Star tick. And then below those images of the adult, female and male, the nymph and the larvae, are the different diseases associated with those tick species. And this is not a comprehensive list, this is just those that are most commonly associated with those tick species. The second exposure factor that we know is very important is the duration of feeding. Very specifically, how long a given tick has been on a person and fed on that person or pet or a companion animal. And the reason for that is, and I'm going to show you the biology of this in a couple of slides, but the transmission of pathogens, those germs that move out of the tick and into you, doesn't happen instantaneously. And it's important to remember that tick feeding is not analogous to a syringe injection. It's not that the tick is just depositing itself as if it were a bag of fluids that's going into you, that there are actual physiological changes in the tick and the germs that have to take place before those germs move out of the tick and into you, and less of interest to us perhaps out of a wild animal and into that tick. That's a whole process, a whole biological process. And because that's a process that takes time, and so we say it's very important exposure factor to know how long a tick is fed. So you may have heard that before, but I want to reinforce why that's important by explaining why it's important, what the underlying biology of it is. So I show this little cartoon here. So this is a tick. She's attached. You can see the yellow here where she's cemented herself in place. She's inserted her straw-like mouth parts into the wound, and she's created a feeding lesion down here, a pool of blood, which I've shown by this reddish area. And what I want to do is show you a cutaway of what's happening inside the tick's gut. So here's a cutaway of very cartoon fashion what the inside of a tick looks like. The first thing I'll point out is that there's a midgut. So this is the belly of the tick. This is where the digestion is going to take place. And I'm just showing one half of the tick. This is actually bilateral, but I'm just showing one half of the tick and giving you a cartoon image of that midgut. The other feature I want to show you is the salivary glands. So the salivary glands are involved with the feeding process, although they're more involved with the effluent, the stuff that comes out of the tick into the feeding lesion. And what I want you to understand just in this image is that those are separated by membranes. So those are very distinct organelles within the tick, organs within the tick, and that they're separated by these barriers. So if we imagine what happens when a tick bite occurs, the blood comes into the midgut, facilitated by stuff that comes out of the salivary glands, but the flow is really unidirectional. The blood goes into the midgut, and fluids from the salivary glands flow out of the salivary glands. And the pathogens that are in the host move into that midgut. Notice they don't move into the salivary glands, they move directly into the midgut. And if we look at the timing of this, if this was a nymphal tick, it would feed in the summer, and then that pathogen would lie dormant in the midgut for basically the time that it takes that tick to digest that blood meal, through the autumn and the spring, the winter, and into the spring of the next year, until that tick is ready to feed again. And then, and only then, when the tick feeds again and takes a new blood meal, does that pathogen migrate from the from the midgut into the salivary glands, where now it's ready for the flow out of that tick. That's the critical piece of that the timing here, that it's only once the tick is fed, and only after it's fed for an appreciable time, that that pathogen moves into the position where it can come out of the tick and back into the host. It's for that reason that we say that it's fundamentally important that the tick feed for a length of time before pathogen transmission takes place. So as a risk factor, we can look at the size of the tick, because it turns out that that we can look at time series of tick feeding, and we can look based on the size and shape, and to a certain extent the change in pigment of the tick, how long the tick is fed. And the longer a tick is fed, the more risky the tick is of transmitting pathogens, if it's going to transmit. And so on the top is just the time series for nymphal ticks, and then the bottom is the course for adult ticks. And usually we say for Lyme disease, somewhere in the area of 1.5 to 2 days is when it becomes critically important, and when the transmission of Lyme disease is most likely to occur. Now, some people will push back, and then they'll say, no, it happens much sooner or later in some cases. Well, it's important to understand this is a distribution, and this is sort of the mean time is 1.5 to 2 days. This is just showing that we can very carefully take measurements of the body of the tick to determine how long it's fed. So 3 exposure factors. The first one is the tick species. The second one is the duration of feeding. And then the third one, of course, is the infection status of individual ticks, because not all ticks are infected. Even if a tick is the right species and it is fed for a sufficient length of time, it may or may not have been infected. So for things like Lyme disease in deer ticks, about 50% of the adult ticks are infected. For things like Rocky Mountain spotted fever in dog ticks or brown dog ticks, the infection rates are much, much lower, probably less than a couple of percent, and depending on where you're talking about geographically. So the infection status is fundamentally important, because infected ticks can infect people, uninfected ticks cannot, all right? So unfortunately, very often these exposure factors don't get measured directly. So I want to speak to, this is how we in our laboratory determine whether a tick is infected or not by using a method called PCR, polymerase chain reaction, that actually detects the nucleic acid of the associated pathogens, either bacterial or viral or protozoan. So I think we pause here for a moment, Jana, for questions.
Yes, and we actually do have a couple of questions. So Kenneth Smith asked, aren't Powassan and some other tick-borne diseases transmitted more or less instantaneously?
Yeah, so there's some evidence in the laboratory that Powassan virus can transmit in about 15 minutes of feeding. How long on average it takes place in a human host, we can't say for sure, but yes, it can have, some of them can transmit quicker than Lyme disease.
Okay. Carrie Conklin asked, is it true that if you disturb the tick, the pathogen transmission can also take place? I believe I heard this from another tick seminar.
You didn't hear that from my seminar, because no, it cannot occur. Transmission of pathogens does not occur once the tick has been killed. So we always tell people that you should remove the tick as quickly as you can. And if you break it off in the process, that's okay, because once that tick is dead, it no longer can undergo those physiological processes necessary for disease transmission. You still have risk of infection because you've now you have a break in your skin and you want to surface sterilize, but you stopped the biological transmission of pathogens from that tick-borne pathogens.
Great, thank you. And Kasia Helgert asked, could you speak to the transmission time of Powassan disease by a deer tick? Is that?
Yes, so we touched on that one. So that's the 15 minutes.
The same, okay, great.
Yep.
Okay. And Deb Moreau said 50% of adult female black-legged are infected, or of both sexes?
50% of both sexes are infected, but male deer ticks, adult male deer ticks don't feed. So they're less relevant to transmission. So it's, again, an exposure measure.
Okay. Elizabeth Dabeck said, do we know if pets can transmit pathogens to humans, and if so, which pathogens?
So there was a question above there about, do ticks removed seem dead?
Yeah, I wasn't quite sure. Actually, Michael Lovell, maybe you can retype that question, because it's not really clear to me what that question is, so I skipped over it.
Do we know if pets can transmit pathogens to humans? Most pets do not serve as very good reservoirs for the pathogens, so it's not like the tick can pick up the pathogen, but also the feeding process takes not only days for an individual host, but then the digestion before the tick is ready to feed takes months and months. So it's not like a tick is going to feed and then feed instantaneously on you. And usually a tick that's fed and then falls in a domestic situation, the tick is probably going to die within the house, because our houses are generally not suitable for their habitat. Okay,
great. All right, well, we have more questions, but I think we'll move along, and we have other question breaks. I've noticed that a couple of people have raised their hands. If you have a question, the best way to do it is to go into the Q&A feature. So if you scroll over your screen, the bottom or top bar will appear, and there's a little option there for Q&A. If you click on that, that's the best place to put a question. It's hard for us to keep track in the chat or with raised hands. So if you folks have questions, that's the way to do it. So I'll let Stephen carry on with talking about tick reports.
So thank you. So I've presented you the 3 ways for measuring tick exposures, and what I'd now like to tell you about is very specifically how we allow individuals and populations to measure their tick exposures through something we call TickReport. So TickReport is a service that we offer through our lab at the University of Massachusetts. It's a fee-for-service, I should emphasize, where we test ticks that people send to us. So human-biting ticks that can be tested for the presence, absence of a variety of different pathogens. And we assess each of those different risk factors: the tick species, the length of tick feeding, and the infection status of individual ticks. And the notion here is just quite simply that every tick-borne disease starts with a tick bite. And so we think it's fundamentally important to have that insight about the tick, about those individual exposures. So we test deer ticks for a variety of pathogens most commonly associated with that species. And then for non-deer ticks, that's mostly Lone Star ticks and dog ticks, we test for the groups of pathogens associated with those other species. So there's just a list shown here. This is just to give you an idea what it looks like. So people send us the tick, we photograph it dorsally and ventrally. In this case, there wasn't much of the tick left that was taken off me. It was actually off my back. And this is the kind of report that we give back to people. First of all, we test the DNA quality. So we determine whether the tick is sufficient quality for us to be able to even do the test. That's what's indicated by the past here. And then you see down below that we test for a variety of different pathogens. For Lyme disease, Borrelia burgdorferi, we actually do two tests which confirm the infection status of those ticks. I won't go into now. If a question comes up why we do that, I can answer that later. But I'll also point out that we capture information about the customer. We don't share any of that information with any source. We just capture it so that we can communicate back with people and get the results back to them. So what we argue is that this is allowing people to make some estimate of their own risk. We don't claim that this is an infection, a diagnosis of disease. And this is something that's, we've kind of gone back and forth with CDC and some other groups about it. We're not suggesting that you use tick testing to diagnose your disease, but we do think that tick testing can be a valuable way for you to assess your exposure risk for reasons I just outlined. There's really no other way to measure exposure except by looking at that tick, because that is the exposure factor. And so in the end, you have information about whether the exposure you had was a positive one or a negative one. And when we look at these data in aggregate, they become of relevance public health-wise because it basically constitutes this passive surveillance. And I think some 76% of you said you didn't know what the passive surveillance was. So I'm very happy to be able to tell you a little bit more about what that is. So it turns out that every single tick, for example, mine that I just showed you, that's sent to us, goes into this public health surveillance network that we make freely available at TickReport.com/stats. So you can go back and you can check on ticks that we've been testing since 2006, both as individuals, or you can look under states, or you can look under individual years, or some crossover of years and state or zip codes, and you can find the results that have been that we found from ticks that we've tested for people through that period. And what I'd like to do is just go through some of that, show you the findings of that passive surveillance and why it gives us a particularly unique view on the risk of tick-borne diseases. So before saying that, let me just tell you how before we started doing this, and there are other groups that do this, Connecticut Agricultural Experiment Station does a particularly good job at it, that before we started looking at passive surveillance by looking at human biting ticks, there are basically two ways that we surveil tick-borne diseases. One was by field sampling of ticks, which means you go out to the field, you collect the ticks by a process called dragging or flagging, and then you bring them back to the lab and you determine whether they're infected or not. And you can see that that really only gives you an indirect measure of the rate at which people are encountering ticks, because you're not looking at ticks that are biting people, you're looking at ticks that jump on a flag when you're really trying to capture them. And so it's not a direct measure of the encounter rate. It does give you a good measure of a direct measure of the infection status because you have the tick there, but it doesn't tell you anything about the duration of tick exposure or how long ticks are feeding on people in a given area or a given circumstance. Likewise, if you look at human case reports, that's the kind of data that the CDC reports, you don't get any information about tick encounter rates. You just assume that every individual that reports positive for disease presumably had a tick encounter, but you don't know if they were bitten by 1 tick or 10 ticks or 100 ticks. It also doesn't give you any information about the infection rate of ticks, because again, The infection could have taken place after a single tick bite, or it could have taken place after multiple infectious tick bites. And then obviously no information about exposure. But what we're doing, the passive surveillance that I'm going to tell you about, it gives direct measures of all these different exposure factors. And we think in that sense alone, it's invaluable. So the first two measure hazard and risk, and our approach actually measures the exposure, which we argue is the thing that is at least as important as hazard and risk.
Great. We have a lot more questions. So Michael clarified his question. He said when he removes ticks, they appear to be dead. And why is that?
So there's two things going on there. One is the ticks may actually be dead. So it turns out that some ticks don't feed particularly well on some people and some hosts, and so they may actually die. There may be something, antibodies in your blood or other things that the tick died before it fully fed. The other thing is once a tick is fed and is replete, it's quite large and it moves very little. It still will show some movement, but it can crawl, but they may be just very less active because they're in the process of digesting that blood meal as opposed to crawling around. But in very many instances, the ticks may in fact be dead. And particularly if a tick is on a pet that you treated with acaricide, topical acaricide, the tick did probably die during the feeding process and remained attached to the dog at that time.
Okay, great, lovely, thank you. Somebody asked, if the tick is squeezed during removal, can the live tick regurgitate?
No, so ticks don't regurgitate. And if they did, they would regurgitate the contents of the midgut, not of the salivary glands. But they actually have a blockage, and a pharyngeal blockage that prevents the regurgitation process. So the idea of squeezing a tick and squeezing the contents out is not biologically sound.
Yeah, great, thank you. Another person asked, have you looked into RNA of ticks incubation period, and are there genes that control transmission?
There are genes that control transmission. There are also factors in the host. So that migration that I talked about a moment ago, where they migrate, the Borrelia, for example, migrate across from the midgut into the salivary glands, that movement is facilitated by proteins that come from the blood meal. So without getting into the details of it, there's proteases that are absent in the pathogen. And so they co-opt proteases from the blood to migrate across those membranes and to cut into those different tissues.
Okay, great, thank you. So I think we have time maybe for one more in this break. Jules Gunenthal said, you showed a quick slide that showed how you can determine how long a tick has been embedded. Could you share a bit more information about this? I think that was the one where you where you showed the size increasing.
Mm-hmm. I'd just like to point out that that's not how long the tick is embedded, that's how long the tick is fed. And I just make that distinction because a tick could remain embedded for a period that's different than its feeding rate. So what we're looking at is how long it is fed, and its body changes shape and size as it feeds. So I'm not sure what more information you're looking for, but if you go to, so those images are from tickencounter.org, which is Tom Mather's page at the University of Rhode Island. And you can see the images that show how the ticks change size and shape over time as they feed.
Okay, great, lovely. All right, we'll move on. And we do have time for some more questions at the end. And a couple of the questions that you've asked may get answered during the rest of the presentation. So
great, thanks. Okay, so told you about exposure factors, told you about what TickReport is. I'd like to tell you now about the passive surveillance database that we've accumulated from an aggregated, I think, close to 80,000 tick reports that we've collected since 2006. That's human biting ticks, their species, their duration of feeding, and their feeding status, which constitute this passive surveillance database. And let me show you the things that sometimes get confused by not looking at the actual encounter rates themselves. So for example, this is a CDC map showing the distribution of black-legged ticks. You notice that are located throughout the southeastern United States. They're found up in the New England, and then there's a second focus up in the upper Midwest. If I superimpose on this map of the distribution of black-legged ticks, if I look at the distribution of Lyme disease cases, you see something quite striking, that Lyme disease is present here in the Northeast and here in the upper Midwest, but very much less frequent, I'll say, in the southeastern United States than it is in the Northeast. And that's for a couple of reasons, one of which is the ticks found here in the South, first of all, are not infected with at the same rate that ticks in the Northeast are. And they also tend not to be human biters. So whether it's behavioral differences or whether they, in fact, as in the past, have been proposed to be a separate species, remains to be seen. But clearly the tick encounter rate in the Southeast is different than the tick encounter rate or the pathogen encounter rate as well in the Northeast and the upper Midwest. So it's important to know what those exposure factors are. So in TickReport, we capture not just information about the tick. So these are all fields that we basically identify. We identify the species, the stage, the life stage, that is, the larval, the nymph, etc., the sex of the tick, the feeding. But also we asked people to tell us what host it was taken from. Was it taken off a person, a child, male or female, the age? And again, we treat all this anonymously. We just get this information for purposes of looking at the exposure rates. And then there's all this client information that we collect, which is really just to maintain communication with the subscriber themselves, the person that has ordered the tick report. And then we capture information about the bite, whether the tick was attached to the skin, where it was attached, was there a rash, etc. And so I want you to understand that we collect these data so that we can do these summaries of the ticks that are sent to us for testing. So this is a little bit, as I say, it's close to 80,000. We've had another big year this year. The ticks come from all 50 states. They're not all deer ticks. In fact, we see a variety of different species, as you'll see in a moment. We do tend to see a large number of ticks come from Massachusetts, and a large percentage of the Massachusetts tick come from Cape Cod. Part of that is just because the service itself is grown by word of mouth, and so a lot of people on Cape Cod are aware of it and have utilized the service. There's also a state subsidy for the state of Massachusetts and a local subsidy in Cape Cod, where local governments or local health agencies subsidize testing fees for residents of those areas, and that doesn't occur outside the state of Massachusetts. So the first thing I want to show you in terms of the summary from our passive surveillance is there are 3 canonical species of human biting ticks. They are the deer tick, the dog tick, and the Lone Star tick. And you can see that they constitute the majority of the ticks that we receive. There's also the Western black-legged tick. So that's the tick from California, Oregon, and Washington that's associated with Lyme disease transmission. Also a fairly big number of those. But there's a whole bunch of other tick species, including things like the woodchuck tick, the squirrel tick, the Gulf Coast tick, which are generally not thought to be, and the mouse tick, for example, not thought to be human biting ticks. And yet these are examples. So let me just pick one out, the woodchuck ticks. We've received 157 woodchuck ticks that have bitten individual people. And I show this just because woodchuck ticks, basically to most individuals, in some cases to trained individuals, wouldn't be able to be distinguished from black-legged ticks. They look very, very similar. And so it's important to be able to really get a handle on the species of ticks that are biting people. And I should emphasize all of these IDs that we've done are by DNA barcoding. So they're completely unambiguous. We don't need to rely on the ability of a microscopist to tell one species from another. And I pick woodchuck tick out because woodchuck ticks are ticks that have been very commonly associated with Powassan virus, and yet not thought to be particularly important public health-wise, because while they do have Powassan virus at some frequency, they generally are thought of as feeding almost exclusively on woodchuck tick. Well, here's 157 examples where they fed on people, and we only get that because of passive surveillance. So with the passive surveillance database, we can ask questions about the threats, the associated threats and the risks. We can ask who's getting bit, when they're getting bit, where those bites are occurring, and what disease-causing pathogens are associated or germs are associated with those bites. So let me show you our answers to those questions. So first, who's getting bit? Well, interesting enough, there's an age distribution. So this is a bit of a busy graph, but the bars show the distribution, the age distribution of nymphal and adult ticks and total ticks in these age categories, less than 5, 5 to 9, 10 to 14. You can see there's a bimodal distribution. So very young children are getting bitten, not so much the 20 and 30 somethings, but then the middle-aged folks like myself, we're getting bit with a higher frequency than the people in this group. And the purple line, that's Lyme disease incidence. So that's the number, that's the age distribution of Lyme disease. And so it turns out that lots of young people get Lyme disease, lots of middle-aged people get Lyme disease, and then less so in this 20 to 30 something group. So this tells us a couple of things. One is it tells us that the reason that those people in that 20 to 30 something group are getting Lyme disease at lower incidence is because they're getting bitten by ticks less frequently for one reason or another. And that was, we speculate why that is. Maybe it was just the degree to which people are going outside in those age groups. But it also tells us that the passive surveillance database, indicated by these bars, which are the exposure risk, is really reflective of the human risk, the human disease incidence. They really do, they measure basically the same thing in this case. So when are people getting bitten? I'm going to go through just 3 slides in succession, and I'm not going to give you a test on this, but I just have to move the screen out of the way so I can see what you're looking at. So here's the 2017 data for deer ticks, dog ticks, and Lone Star ticks. And if you just keep that image in your mind's eye as I scroll into 2018, you see there's a little bit of a shift, a little bit higher peaks maybe in some place, a little lower in others. And when I go into 2019, another little shift again. So I summarized these data before 2019 was done. So this continued into the fall. And I show this just because I want to say that the seasonality, the predictive seasonality of ticks is pretty solid, that every year, despite the fact that we every year get calls from newspapers and local TV news and sometimes national news about whether it's going to be a big tick year or not, based on the ticks that we see biting people, there's some constancy to it. There's some relatively uniform rates at which people are encountering tick bites. In some cases, they come a little bit earlier in the year, some cases a little bit later, but a lot of that has to do with the weather and whether people are getting outside or not. And so sometimes we'll see if it's a particularly wet spring, and people aren't out hiking to the same extent that they are, then we'll see a slight shift in the peak. But nonetheless, the ticks come every year. And I think that's important for people to understand that the risks, while they're one thing to point out, so let me go back to 2018 because the year is complete, that for deer ticks, there's really never a zero risk. But if you wanted to pick a time to go out and have a picnic, you'd want to be in this area or this area as opposed to this one, or at least be more aware of the fact that the risks are higher in May and June for deer ticks. And on that subject of deer ticks, so all of these data, by the way, are based on ticks, that human biting ticks. It's not just the distribution of ticks in the wild. This is the rate at which people get bitten by ticks, in this case, in the state of Massachusetts. And so these are just deer ticks now for 2017. And if you look, you'll see that there's this bimodal distribution is attributable to adult ticks. So there's a spring distribution of adult ticks and a fall distribution of adult ticks, coupled with a late spring, early summer distribution of nymphal ticks, and then a late summer distribution of larval ticks, which you may not see down there. A couple of things about larval ticks. One is they're not infected with many of the pathogens that are of biggest concern to us, for example, Lyme disease. And one of the reasons they may be so low incidence here is that they're so small, they may very often go undetected. nonetheless, we do receive a few every year, a few human biting larval deer ticks. The thing I like to show while I have these graphs up is notice that in August, September, October, there's really a cessation in human tick biting activity associated with deer ticks. And I'm always amazed at the number of people, friends or family members that will tell me, boy, really hasn't been a bad tick year. And they're talking about August or September. And the August or September low tick burdens they're seeing, that's an annual thing. And what I think people often mistakenly do is they think they're comparing August and September with August of September of the previous year, or sorry, May, June of the same year, as opposed to August, September of the same year, of the previous year. August, September, there's always a low number of ticks. May, June, there's always a high number of ticks. And so I'm just surprised at how many times people forget this sort of every September and August, September, October period. Really, we see a lull in the activity associated with human biting deer ticks. Just a reminder of the pathogens that are associated with the different tick species. I'm going to show you in a moment the distribution of those pathogens in the ticks that we test. Where are the tick bites being reported? Well, we say that in the state of Massachusetts, virtually every town of the 351 towns in the state of Massachusetts has deer ticks or has reported deer tick bites. It's becoming true for it's certainly true for every county in Massachusetts, and every county in the state of New York. And I know there's an active surveillance program that the state of New York does where they look at flag ticks as well, but we can also report that there are human biting ticks in each of those counties. But another way of asking the question of where the tick bites are occurring is looking at where on the body they occur. So again, we think there's some public health value to this, to say that 18% of the ticks that are sent to us come from the upper leg, 13% come from somewhere in the hairline. We think this is useful information, for example, for parents that may be recently moved to a tick endemic area, to know where they can look for ticks on their kid. Because just backing up a few steps, if you get the tick off early, even if the tick is bitten, but you get the tick off early, you can greatly reduce the risk of many of the most common tick-borne pathogens. The other value that comes from looking at ticks that are biting people, as opposed to looking at disease in people and or looking at ticks that are just in the wild, is we can determine the distance between where the tick encounter occurred, so where the tick bite occurred, and where the person lives. So here, and I'll show you why that's important in a moment, but we find that most people, you can see some close to 40K here, report tick bites probably pretty close to their home. But there are certain numbers of people that get bitten by ticks that are quite distant from their home. And this is important because, so this graph shows this. So each of these little arrows shows the distance between the place where the tick bite occurred, and then the arrow points to the place where the person lives. So these are all tick bites that occurred in the Northeast, and then the arrows point back to where the person actually lives. And so we capture both those location points, where the tick bite occurred, where the person lives. And the value in that is, let's take this one. So this is the one tick that we received from a Hawaii resident. This was a Hawaii resident that had traveled to the state of Massachusetts, Cape Cod actually, been bitten by a tick, returned home, found the tick on themselves when they returned back to Hawaii, send it to us for testing. Had that tick not been found, and the first person reported back to their doctor that they had Lyme, or reported to their doctor signs and symptoms associated with Lyme disease and been diagnosed with Lyme disease, very likely that would have been diagnosed as a case of Lyme disease that was acquired in Hawaii. But point of fact, it was acquired by a tick that had bitten the person in the state of Massachusetts. Now, it may be that they remembered their travel history and reported that back to the doctor, and the doctor would have reported it out properly. But we're sure there are instances
of people being bitten in one part of the country and then reporting their illness in another part of the country, which really clouds the surveillance picture substantially. And it happens both directions. So there are people that can be encountering ticks on the West Coast, in this case, California, and then traveling back to their home, for example, in the Northeast or the Southeast, and reporting the incidents there. These are all cases of ticks that were sent to us. And we had that hunch that that might be happening. And so in 2019, we actually published a summary of about 400 ticks, a little less than 400 ticks that we had tested that were sent to us from the West Coast. And what we found was, curiously enough, most of the ticks that were positive, most of the human biting ticks sent to us from the West Coast, California, Oregon, and Washington, that were positive for Lyme disease, were actually from non-endemic ticks. In other words, ticks that aren't naturally occurring on the West Coast. And when we followed up with those individuals that had been bitten by those ticks, each of them had a 2-week travel history to the Northeast or the upper Midwest. So most of the ticks that, I think I have it highlighted, oh, sorry, I don't. Most of the ticks that were in this study, which is all the ticks that were sent to us for TickReport from the West Coast, that were positive were actually ticks that came from the Northeast. There were ticks that were endemic as well, that had a low level infection, but you can see almost double the infection rate in the Ixodes scapularis is the black-legged tick, which is endemic to the Northeast, not found in California. So that's why we think it's fundamentally important for surveillance purposes and for risk assessment to look at the human biting tick and to capture this information about where those tick bites actually occurred. So one other thing I'll talk about is the red meat, the Alpho-Gal red meat allergy, or sometimes called Alpho-Gal syndrome. So listeners may be familiar with this. There's an allergy to red meat that's induced by the bite, thought to be induced by the bite of a Lone Star tick. And this map shows the distribution of reported incidences of red meat allergy. So I forget, now that I look at this slide, what these percentages, clearly not 37% of the population. This is, I'm sorry, I'm forgetting what this slide is. It's not my slide, it's from a site that reports, I think this is the distribution of the total number of Alpho-Gal syndrome cases. 37% of all Alpho-Gal syndrome cases are found in this range. Viewers might want to correct me if I have that wrong, but what I want to show here is that this is all based on human reported human cases, whereas when we look at our data, we find a slightly different distribution of Lone Star ticks, and we think that this is probably the place to start to look at the risk for things like Alpho-Gal syndrome. If the tick bite is the principal risk factor, we really ought to be looking at the human biting rate by Lone Star ticks. So that's the distribution nationwide, and this is in the Northeast. And one of the reasons we think that's important is that typically Lone Star ticks were found here in the Southeast, but we now know they're expanding their range. And in fact, we receive more ticks through TickReport from Long Island, more Lone Star ticks from Long Island than we receive black-legged ticks at this point. They've also established themselves on Cape Cod. There are year-round populations. And so risk for things like Alpho-Gal syndrome are likely to be likely to have an expanding range for those risks, and it's important to look at the ticks to be aware of that. Just summarizing what I said a moment ago. So one of the beauties of being able to receive the ticks that are sent to us locally, for example, here in Massachusetts, so here's Centerville, Massachusetts, which is a town in Cape Cod, and then these are the data for the state of Massachusetts, is that when people ask us questions, I think I saw some of them scroll by. I know we're going to get back to questions in a second. But when people ask questions about infection rates, rather than tell them national or statewide infection rates, what TickReport is allowing us to do is give a very detailed distribution rate for different pathogens locally. So we can tell people, based on the number of ticks that we've looked at from a town or a zip code or a county, what the infection rates are. And for things like Lyme disease, I can tell you that wherever deer ticks are present, wherever black-legged ticks are found, that 50% infection rate is a pretty solid number for adult ticks. It's somewhere 25 to 30% for nymphal ticks, but wherever those black-legged ticks are found, that's basically going to be the infection rate. That's not true for things like anaplasma and babesia and some of the other rarer pathogens. In some cases, in coastal regions, those pathogens might be upwards of 10%. So 10% of the black-legged ticks might be infected, for example, with babesia. Whereas when you move further inland, you'll find in some cases 0%, and that's biologically meaningful differences. And so TickReport.com/stats, that link down in the lower right-hand corner, can be used to go and actually look at what's been sent from your zip code. And it's by no means comprehensive, because we know there's still a bias in the regions that we receive ticks from, but it does give you a real hard data about the pathogen distribution in the areas that y'all are living. This is just another view of it showing the distribution. And I use this to show you that that Centerville may or may not be representative of the state of Massachusetts for all these different pathogens. And you can go and look at these. You can make comparisons for New York and individual counties, or Vermont, individual zip codes, and et cetera. So rather than try to summarize these data, which are also available CDC through the active surveillances, you can look at the passive surveillance data and get a handle on more localized occurrences of these different pathogens. Another value of the passive surveillance is because the ticks are sent to us. So one of the costly things, literally costly things of active surveillance is you have to send staff out to collect the ticks. In the case of passive surveillance, the ticks are sent to us. So we get a broader geographic distribution and a broader host range than other active measures. And so this is a summary. This is another paper that we wrote back in 2018, where we had identified another association between a particular Ehrlichia species and those Ixodes cookii ticks, the woodchuck ticks that I told you about before. They're usually found in woodchuck burrows, but we actually found potential human pathogens in those as well. That was made possible because the ticks were sent to us from that region. So just to finish, I want to just come back and say that, while I think most state health agencies, certainly here in Massachusetts, it's become recognized that passive surveillance has an important value. CDC is somewhat less enthusiastic about it, whereas the passive surveillance itself has been accepted. The CDC party line on the individual benefits of tick testing are less positive. And so I just want to speak to that for a moment. Their warnings are that they want people to understand that this is not a diagnosis, and they're very much correct in asserting that there's a concern that if someone sends a tick in and the tick tests positive, people should understand that that is not a positive diagnosis because of all the reasons I told you at the beginning of the talk. Positive ticks have to feed for a length of time. They have to be the right species, et cetera, et cetera. So if you just have one of the risk factors, it doesn't mean that you got infected, and by no means should that tick test serve as a diagnosis. On the other hand, there's also concern that if the tick is negative, you should not be led to believe that you were not infected, because while you most certainly were not infected by a negative tick, you could have been infected by another tick that you didn't detect on your body that never got tested. So for those reasons, the CDC is somewhat staunchly negative about tick testing. But we think that it's of value to have this information with those caveats. It's a value to have this information so that you get some measure of risk, that absent the tick report, you have to basically, in some cases, rely on just information about just knowing that a tick bite occurred and trying to remember what actually happened. So this is a flow chart from Alan Steer showing different courses of action after certain summer events, like a tick bite occurs, or you get a skin rash, or you have a summer flu. And notice they say the no test here is the test of the human patient. So you can say there's no tick bite, there is a tick bite, don't bother testing. You could just go to an oral therapy. Or there's a skin rash. You don't have to test. You could just go to an oral therapy. Or in the case of a summer flu, no test, or test if you wish and go to oral therapy. Our contention is that there's got to be some value to having that risk assessment to mitigate that, to have another piece of the puzzle before making that determination of whether to treat prophylactically or not. It's not the end all be all. It's not a diagnosis. It's not meant to replace diagnosis, but it is. It's hard to say that it's not of value in terms of risk assessment. So I bring that up because many people ask the question about why the CDC that recommends against this, why? So I thought I'd pre-answer that question. The other thing is for the entire left hand of that diagram, of course, pertains to transmission of Lyme disease. For non-Lyme pathogens like Anaplasma, Babesia, Rocky Mountain spotted fever, in some cases, the familiarity on the part of the docs that are out there is lower. And so it's sometimes valuable to just have that risk assessment to get it into the clinical picture, to know that, yes, the tick that bit someone that's experiencing fever was negative for Lyme disease, but it was positive for Anaplasma. And so perhaps the fever is associated with the Anaplasma, and that could be put into the diagnostic picture. Not a diagnosis, but a risk factor that can be used in assessing a clinical diagnosis. And we have growing number of anecdotes and at least one case report where we've shown how tick reporting has led to that kind of insight. So this is a case of a child that had been bitten by a tick that was Borrelia miyamotoi positive. Borrelia miyamotoi is a relapsing fever-like spirochete. And through the benefit of having tested the tick, the doctors were able to get to the diagnosis more quickly than they would have. In fact, arguably may not have got to the diagnosis of this very rare tick-borne pathogen had it not been for the tick test and one of the parents that sort of pushed the docs towards eventually testing the child for the same pathogen. So I'll say just in closing that we've been cognizant of the fact that we're filling a gap because we're doing this passive surveillance, we're giving people individual risk exposure information, but we're also answering questions all the time. So we have on our website where you can order the tick report, we have FAQs, frequently asked questions. We run a chat line that's down in the corner here where you can ask questions in real time of a live person. So we have a staff that can answer questions, not just about tick testing, but about tick bites in general. In some cases, help people to identify ticks by a species, by a picture. And in some cases tell people that they weren't bitten by a tick. So we do receive a fair number of sesame seeds or beetles or scabs that people pull off themselves. And we can tell those people, this is not a tick bite. You should, you don't have to worry about this particular instance. The statistics page, in case you didn't jot down that link, is also available. So if you go to TickReport.com/select statistics, you can look at those downloadable passive surveillance statistics and use the chat tool down in the lower right-hand corner if you had questions about things I talked about today or you have questions that we talk about in the future. This just shows you the number of conversations. This is through April of 2019, last year, January to April 2019, we had about 5,566 conversations with tick bite victims, that some of whom sent ticks in for testing, but some of whom were just pulling a tick off the kid in the middle of the night and they wanted answers to questions. And our turnaround time is 4 minutes and 23 seconds. So if you send us that at 4 o'clock in the afternoon, you would get a real-time response. If you sent it at 4 o'clock in the morning, it would probably roll over to an email and then you'd get a response back to us from 8 a.m. But on average, we're getting a pretty quick turnaround. It's free service. We do it to help people out. And I'll close there and just say that this is a big team. This is a team effort. Myself and my associate, Dr. Guangshu, and the team of people that run TickReport. These are mostly full-time technicians in the front row. There are a few undergrads that sort of fill in, but it's mostly full-time research techs that run the service and provide the service that we give out. So I'll turn it back over to you at this point, Jana.
Great, lovely, thank you. That was really informative, and we have a lot of questions. So we may not be able to get them all answered, but I will ask some of them, and I will focus on the ones actually that use Dr. Rich's expertise, and because some of the other questions may get answered in other webinars. So Beatrice Center asked, do you pull data with other labs like UMaine or Connecticut Ag Extension Station, Experiment Station, to supply pathogen infection risk accurately in those locales? And if not, could you?
Have we pulled data? I'm sorry, I should read the question.
Do you pool data with other labs?
Oh, we don't pool data. We have made so the U.S. Army tests ticks. So we have pooled data with the U.S. Army because we managed to coordinate methodologies with them. But Connecticut Ag Station uses different methodologies. They don't really disclose exactly what they use. We publish all of ours, by the way. They're all freely available. Maine uses our approach. They basically came to our lab to learn how to do tick testing, but they don't share any of their raw data presently. So we have spoken with groups before about getting aggregated data, but there seems to be a bit of, we have some work to do there.
Okay, great. And she also asked, is it expected that non-endemic ISCAP, and I think that's an abbreviation, will or could become endemic on the West Coast as its range continues to expand?
It's not likely that Ixodes scapularis would expand its range to the West Coast, no.
Okay, all right, great. And Kristen asked, do you know if there's comparable data or comparable reporting or agencies available in Canada?
There is so we've tested ticks from Canada for a number of years, and now there's a testing service in Ontario, I think, that's, at least for the province, it tests ticks. I don't know their address, but.
Okay, great. Thank you. All right. What are the factors that account for the low percentage of bites in August, September, and October?
Basically, it's because the larval ticks are less human biters. They tend to be lower in the leaf litter. They bite on mice, and they probably also bite at rates higher than is reflected by our submission rates, and they go undetected.
Okay. Great. And actually that kind of half answers somebody else's question. They were saying, can all life stages of black-legged ticks transmit? No.
So larval ticks the larvae are hatched from eggs without infection with Borrelia burgdorferi. They can retain other infections as eggs, but they're not infectious. So one of the reasons not to worry about the larvae is that they're not infected with Lyme disease.
John Galhouse had an interesting comment. He said, what's the influence of cost and availability on the samples? For example, are people in their 20s less likely to submit and pay for a sample, whereas parents or middle-aged people are more likely to pay for it?
That may be part of it. Yeah, we don't know. Yeah. When we started doing, I just would like to say that when we started doing this, we didn't have any means to do it. We had to make it a fee for service. And at one point we thought, well, we'll just keep doing it. And if it's important, people will pay for it. And then eventually, if it's really important, maybe that it'll get subsidized. I now think that, having some kind of fee for service is valued because people do value things that they pay for. And also people are getting individual benefits for it. So very often people say, why doesn't the state just pay for this? My experience is that, this is getting a little bit off the question, but my experience is that if the state pays for it, when funds dry up, the program goes away. And so there's something valuable to sort of having it in the market and sort of have it being, by all means, as affordable as possible, but to have it as a fee for service. The consequences are that we know that not everyone has access to it, including people that in the age group that is described, but also in other groups that don't have the money to pay for the tick testing. So we're trying to think of ways to offset those costs.
Okay, great, thank you. So Maida and Nathan said your maps are showing the distribution of, Ixodes versus Lyme incidence are striking. What's known about the spatial variation in density throughout its distribution? Could there be lower densities in the Southeast US contributing to low exposure there?
There are less ticks. There are less ticks to be flagged in the Southeast. So whether they're just more nest parasites or don't tend to come out in the areas that are flagging, they certainly seem to be less frequent there. Yeah.
Okay. Is there a percentage of ticks that die in overwintering?
For sure. Yeah, there's an overwinter attrition rate. It's not really enough.
Mm-hmm. Right. Okay. Ashley Kennedy asked, have you observed an uptick in submissions this year because of the pandemic? And it seems like people are spending more time outdoors.
Yeah, so March was a banner year. Was a banner month. We'd never seen a March like we saw in the March of 2020. And we attribute it directly to the fact that when people couldn't go to work and couldn't go to play and couldn't go to school, couldn't go to school, couldn't go to work, they went to play and they went outside and they got bit by ticks at high rates and they were submitting them to us. It sort of returned to normal levels in May and June, April and May into June. And then we went on the university was furloughed for 5 days, so we got a little break in our data. But yeah, by all means, at the beginning of the COVID-19 quarantine, we saw uptick, pardon the phrase, in submissions.
Great. So going to work keeps you safe from Lyme, basically. All right. Pat Vittum said, why does it appear that there are fewer cases in Massachusetts than its neighbors? That's hard to believe.
Hi, Pat. Pat's a friend. Fewer cases of Lyme disease, maybe? I'm not sure what we're...
That's what she doesn't actually indicate that. It says fewer cases in Massachusetts, so maybe it indicates Lyme. It must have been from one of your slides.
Yeah,
so
I'm not
Pat,
sure.
I would say maybe if you want to clarify that, we can come back to it. Okay, great. All right, we're going to, I'm just going to ask maybe one more, and then if there's one in this list that I know you can see the list too, if there's one that jumps out to you, feel free to answer that one. Stephen Bramer asks, any illogical or chemical control research in your lab?
Any which kind? Sorry.
It looks like iological, I-O-logical, or chemical, or hemicel, it might be, control research in your lab.
Hemosil, maybe?
Yes, it's right at the bottom of the list of questions.
Chemical? I'm sorry, I don't know what either of those terms. I'm not familiar.
Okay, good. Well, that makes me feel less stupid for mangling up the pronunciation. So great. And if there's any one of these questions that you can see here that you think would be helpful to answer, please go ahead and do that, and then we'll go on to the end of the presentation.
Well, let me pick out Emily Landry's question. So she says, is it true? Is there supported evidence that Lyme disease originated in Lyme, Connecticut? So I would speak to that and say that Lyme disease as a reported phenomenon started in Lyme, Connecticut, but we by no means think that Lyme disease originated in Connecticut. We think that it was probably around in the Northeast. It was in Europe, for example, 100 years ago. And so as a epidemiological phenomenon, it was first detected in Lyme, Connecticut. That much is true, but no one has ever really proposed that it started in Lyme, Connecticut. And the reason I picked that one to answer is there is a book that's been written that has proposed that this is actually the consequence of a biological warfare experiment gone awry, that this was released from, I think it was a Plum Island story. And I just say that others have written about this extensively. There's very little evidence for that being true, that this is, it makes a great conspiracy theory, but there's really no evidence that this is not anything other than a disease that's been in ticks and in rodent populations for millennia, and that ecological circumstances have changed and we have more deer and more ticks than we ever had in the past, and so we have more disease, but that it's probably always been there. And the real evidence for that is the global distribution, that the pathogens are throughout the whole Arctic, they're across Eurasia, from Siberia through Northern Europe. And so it's just not parsimonious that would have been attributed to an accidental release of a biowarfare weapon.
Okay, how interesting. Thank you. So All right, wonderful. And for those of you who have other open questions, hopefully they'll get answered in other seminars. I appreciate Stephen being so patient with answering many of the questions that we did. We have some questions for you. I want to follow up on the original poll that we gave you. And I think there are 7 questions that are going to pop up on your screen. And hopefully there we go. They should have popped up on your screen. And I ask that if you could just take a couple of minutes just to scroll through and give us some feedback on your rate of knowledge of different areas. That would be really helpful for us. We use it for making our webinars better and stronger, but also in seeking funding to provide these and make them available. So I'll be quiet, and yeah, we have to source some Jeopardy music. Okay, so we have about a 75%, actually just went up 76% rate, and we'll share the results with you. There we go. So you'll be really pleased to see this, how now majority of people are very knowledgeable about blacklegged tick feeding process and about the timing of disease transmission. There was some good questions about that too. That was really interesting. And approaches for assessing risk of tick-borne disease. Most people are either moderately or very knowledgeable, and we have 6 people who are now extremely knowledgeable. The same about lab testing for both humans and ticks. And then the question that is really great for us, as a result of this webinar, how likely are you to increase your implementation of IPM? And 43% of people are very likely and 21% are extremely likely. So that's really made a difference. And we appreciate you listening so intently and answering those questions for us. And oops, I should, gosh, I had not put this on a slideshow. So the next, we just wanted to let you know that we have a place on our website where if you want to collaborate with someone in the Northeast around IPM, that you can, oh, I'm sorry, it seems to have paused that. Resume share. Hold on 2 seconds. I have to resume the sharing of the PowerPoint. I apologize, something happened. So we have a site called Find a Colleague, and you can post a listing on there of who you are and your research interests. You may be a farmer, an agricultural researcher, a grad student, or anything in between. And it's a good way for people to connect and find each other when they have similar research interests. We have some 4 upcoming, 5 actually upcoming webinars listed on this page here, going through October of this year. And we may add another 1 or 2 to this list, so please feel free to come back and check on our website. The one thing I want to mention to you is you need to individually register for each webinar. Even though it's a series, you'd need to go in and register for each one. And you can find the link on our website of how to do that. So the very next one coming up is in a couple of weeks. Dina Fonseca and Matt Bickerton at Rutgers are going to speak about Asian longhorn tick. And I know Stephen mentioned that a little bit today. And they're going to go into it in more detail. The recording is going to be available in about a week, and you'll get an email from me with a copy of the link to that. And finally, I just want to acknowledge and say thank you to our funding agency, which is NIFA, which is part of USDA. We receive a grant from them, and the Laboratory of Medical Zoology that employs Dr. Rich. And to thank you very much for your presentation today. Thank you for your expertise and your patience and lifetime of experience and dedication that it's given for you to be able to answer those questions and put this together so beautifully. So thank you for dedicating your life to what you've dedicated it to and sharing it with us. I appreciate it. So
thank you, and thank you for the invite.
Great, lovely. All right, well, with that, that is the conclusion of our webinar, and I look forward to seeing you at another webinar sometime this summer. All right, bye-bye.